Okay, yesterday we have discussed how to use the vector, right, to clone the gene and then to reproduce the gene of interest that we clone. And today we will discuss the production of protein from the cloned gene. And then to produce the protein through the cloned gene, then we have to use a specialized vector. So that's what we're going to discuss, right? And then to produce the protein from the clonal gene, so basically you need to grow the cell, all right? So first of all, you of course you clone the gene into a vector and then introduce the vector into the bacteria cell or yeast cell or animal cells, and then you have the mass production of the cell. And then so in this diagram here, just to show you that we clone the gene and then introduce into the cell, and then we want this gene to be expressed, all right, to be expressed, and then can produce the protein, all right. So each cell can produce a little bit of protein. Then so you need to grow the big culture, right? A big culture so that you can collect a lot of cells from this batch culture. Then you have hundreds, millions of cells, and every one produce this little bit of protein. Then you can accumulate a lot of protein, and then you can also isolate the protein from the culture, which is the whole process we will discuss today. And this is a batch culture, and which is normally what we do in the laboratory. Okay, so we do this like day we grow the bacteria or Saccharomyces cerevisiae. Every day just use the flask and then inoculate the star culture so that we can grow the batch culture. Or another way is a continuous culture, which I will show you the video today about the continuous culture. This is like industrial grade. So like in a biotech company or pharmaceutical company, when they want to have the mass production of a protein, then they will do this continuous culture. And this is commonly seen in the biotechnology field. All right, so that's what I will show you in the video later today. And so yesterday we had discussed the cloning vector. So that would be the production of DNA from a cloned gene. And today we will discuss the expression vector, which will be the production of protein from the cloned gene. Okay. And yesterday when we discussed the cloning vector, so I mentioned that three important factors for cloning vector. So one is the multiple cloning site. The second is the origin of replication. And number three is the anti-selectable marker. And for the bacteria, mostly we use the antibiotic resistant gene. And for the expression vector, okay, and three important features that an expression vector should have, okay. Number one will be promoter. Number two, terminator. And number three is a ribosome binding site. All right. So this is a piece of let's say this is the vector, and then you should have a promoter, ribosome binding site, and terminator. And then you're going to place the gene of interest behind the ribosome binding site. Okay. So that's the three important features for the expression vector. Okay, and now why the promoter is important, all right? So because the promoter will control the first step of the expression, because the promoter will control the transcription. If you have a strong promoter, then you will produce a lot of messenger RNA, all right? If you have a weak promoter, then your messenger RNA will be less, and the protein production is based on the gene expression. And then if your first step, the transcription, produce less messenger RNA, then you will not get a lot of protein production, right? And for the E. coli, so we know this promoter, the -10 bucks, -35 bucks, the Tata bucks, and also for the animals, the eukaryotic cell, -25 bucks. So these are the strong promoter, okay? That's a promoter. And because the promoter is so important, so we will also discuss how the promoter control the expression later. Okay, so here just to explain, if you have strong promoter, then you can produce a lot of transcript, and from this transcript, then you can produce a lot of the protein. If you have weak promoter, then you have less transcript, and then you have low protein production. And how do you define strong or weak? The strong promoter, that means you have the almost exact identical consensus sequence, all right, -10, -35. And then if you have not the almost identical, then you weaker the promoter. You have the weaker promoter, right? And for the promoter selection, okay, so basically we have two major type of gene regulation are recognized in E. coli. We can have induction and the repression. An inducible gene is one that the transcription is switched on by addition of a chemical to the growth media, all right? Inducible promoter, that means the transcription can be switched on when you add a chemical to the growth media. Okay, that's the inducible. So the inducible gene for the induction, an inducible gene is one that transcription can be switched on by adding the chemical. Repression, so when you add a regulatory chemical, then you switch off. That's called repression. Okay, and so that the factors that can control the gene expression, we have the cis element. Cis element, that will be the DNA sequence. And most cases, we refer this cis element would be like, for example, the regulatory sequence. It could be the promoter or activator, all right? The promoter sequence, all right, or the activator recognition sequence, activator binding site. And trans element are the protein that will recognize the cis element, okay, and then can either induce or repress the expression, all right? So that's trans element. You have cis element and trans element. And then these are just examples of the gene regulation occurring in the bacteria. So the panel A here is the inducible gene, all right? So inducible gene, that means when you add the regulatory chemical, you switch on the transcription, so you produce the transcript. A repressible gene, that means when you add the regulatory chemical, you switch off. And all this is controlled by the promoter, all right? It's controlled by the promoter. And so basically, here, that's the definition of the messenger RNA regulation. So the binding of a regulator to DNA, all right? The regulator is a trans element, and DNA will be a cis element. So if you have binding, all right, for the positive control, if you bind something, then turn on, then the positive control will turn on the transcription. If no binding, then turn off, right? If a negative control, if you have something binding, something bind to the regulator, to the DNA, then you turn off, and no binding will be on, all right? So here, for the repressor binding side, without binding, all right, without binding, then it's on, the transcription is on. With the binding, then you turn off the transcription, okay? And then for this activator binding side, all right, without binding, here is off. And then if you bind the transcriptional activator, which is the trans element, then you turn on the transcription, okay? So this is the negative versus positive regulation. So This is just an example to show you the lactose operon. The lactose operon, and yesterday we did mention some of the lag operon, the LAC-Z gene, all right, LAC-Z gene, which is this one here, LAC-Z, like the PAC8, the vector, all right. So they have the LAC-Z gene, all right, and so this is the detail of the lactose operon, okay. And the lactose operon, and you can see the promoter is here, the operator and repressor gene, all right? And then this is structural gene, and all these three genes is under one promoter. And because once they turn on this expression, you can express these three different genes and all these three together. So that's why we call this as operon. Right? The operator is the repressor binding site. Okay, the promoter, of course, that's the RNA polymerase binding site to activate the transcription. Okay, so so if we have the transcription of the repressor and then the repressor bind to the operator, then this will block the RNA polymerase to bind to the promoter, and this will switch off the transcription. However, if you have the inducer, all right, inducer that can bind to the repressor, all right, bind to the repressor, therefore the repressor will not be able to bind to the operator. And then the RNA polymerase will be able to recognize the promoter and then activate the transcription. So this is the lactose operon, okay? And then for the transcription, for the expression vector, okay? For the expression vector, a lot of the typical expression vector, that will be the LAC promoter. And the LAC promoter responsible for the transcription of the LAC-Z gene, and so we have the promoter -10, -35, and then the IPTG can be added to induce the expression of LAC-Z, all right? So that's the LAC promoter. And also sometimes we also use the TRIP promoter. The TRIP promoter basically is the responsible for the expression, and this will be responsible for the production of the tryptophan, right? The one of the amino acid tryptophan synthesis. All right, so you add the 3-beta indole acrylic acid, then you activate the transcription. And then if you produce too much tryptophan, then they will have a feedback effect to come to stop the transcription. So that's the TRIP promoter. And also we have the TEC promoter, also for the LAC-Z gene expression induced by the IPTG. It's a hybrid between the TRIP and LAC promoter, all right? So that's a TEC promoter. And also we have T7 promoter that's induced by the IPTG or lambda-PL promoter, all right? That's responsible for the transcription of lambda. These are the typical expression vector, okay? Expression. So that's the importance of the promoter. Control the whole protein production, because that's the first step of a gene expression. That's why it's a critical, very critical. If you don't produce enough transcript, then you cannot get the mass production of protein. Okay? So these are the typical expression vector. And then the fusion protein. So the fusion protein, that means, so basically after the promoter, all right, you can produce the transcript, all right, and then the transcript you have to translate into the protein, okay, and then translate into the protein you need this ribosome binding site and terminate, okay, and the ribosome binding site, basically ribosome binding site is a place where the ribosome bind and then start a translation, and then if you clone a piece of gene right immediately after the ribosome, okay? So that means you will express the completely the gene of interest. However, if you clone the gene a few base pair after the ribosome, the start codon, then you will produce a fusion protein, the hybrid of the bacteria, the vector sequence, and also your gene of interest. That's called fusion protein. So that means that's why I say if the cloning site is not immediately adjacent to the ribosome sequence, ribosome binding sequence, then the foreign gene is inserted into the restriction site and produce a hybrid gene, and that's called a fusion gene, fusion protein, right? So for example, in here, so this is a expression vector. You have promoter ribosome binding site terminator, and then so if you have the foreign gene not immediately adjacent to the ribosome binding sequence, then you produce a hybrid. All right, the hybrid, I mean you have part of the vector sequence and then your gene of interest sequence. And when they translate into the protein, all right, when you clone the fusion protein, you have to be extremely careful because when they produce the hybrid, you can change, for example, in this case, you can change the open reading frame. Once you change the open reading frame, then the protein will be different from your original expected protein. Right? When you design this kind of the gene cloning, you have to be very careful to make sure you clone the gene in the correct open reading frame. And therefore, you can produce the correct protein. All right, so that's the fusion protein. So the advantage of the fusion protein, it cannot be an efficient translation because it reduces the possibility of a secondary structure. Okay, and also I can stabilize the presence of bacteria peptide at the start of fusion protein can stabilize the protein molecule so that it will be not degraded. Also, it can be directed to a correct position in the cell. Also, the most importantly, easy purification by affinity chromatography, which is a very important issue that we will also discuss later in today's class. All right. So these are the 4 advantages for fusion protein: efficient translation, stabilization, and direct the protein to the correct position, and the easy purification. And for the purification, okay, for the purification in the expression vector, sometimes you have to incorporate some specific amino acid, and this amino acid will be expressed and then give you a tag, and so you can help you to purify your target protein. So these tags that include the HIS tag, GST tag, FLAG tag, and T7 tag, and these are just the amino acid that can be recognized by a specific, like for example, T7 flag can be recognized by the antibody, so you can purify it, all right? And GST also antibody, and the HIS tag can also purify through the metal chelation chromatography, right? So these are the tags that incorporate into the expression vector, okay? So later on, I will show a typical expression vector and then to show you how they incorporate the tag into the vector. And so easy purification, because once you have the protein expressed with the presence of tag, then you go through this column chromatography. So in this example, you have this the glutathione S transferase fusion protein. Then you go through this column, the glutathione at aqueous beads, all right? So those proteins will bind to the beads, all right? Bind to the beads and stay here so that all the irrelevant material would be separated from the target protein. And then later on, you can elute the protein. So that's how you purify, isolate the pure fusion protein. Okay. The disadvantage of the fusion protein, that's because the presence of E. coli bacteria segment can alter the property of recombinant protein. Okay, so that's one disadvantage. And there are methods that you can remove. You can remove these bacteria sequence, all right? So to remove the bacteria sequence, so if there is a methionine in the junction here, all right, so the fusion protein, you can use the cyanidin bromide so that you can separate these two segments. They separate the bacteria sequence from the protein. Okay, that's the way that you can use to remove the bacteria sequence in the fusion protein. Any questions so far? Okay, so good. So let's continue. So the general problem with the production of recombinant protein in E. coli, right? So the general problem, so basically there are three problems. So basically, number one, the foreign gene might have the intron, right? If this is eukaryotic gene, so it might have the intron, okay? Because in the bacteria system, there's no intron, and then if you clone the gene directly, then you might introduce intron into the system, and the E. coli messenger RNA, then you will not be able to produce the the gene you want. And in this case, you will try to do you would use a reverse transcription to get the complementary DNA. You would try to clone the cDNA, okay? And so you can eliminate the problem. Also, it can have the premature termination of a transcription because the foreign gene might contain sequence that act as the termination signal in E. coli. And then that basically then the sequence cannot be expressed completely. And in this case, then you will try to use some in vitro mutagenesis to remove those early termination signal. And also there's a codon bias. That gene, because the codon bias, that means those are codon that used in the eukaryotic cell might not be commonly seen in the bacteria cell. Therefore, in this case, you also need to use the in vitro mutagenesis to mutate a specific codon to make it into the popular codon that used by the bacteria cell. All right. So these are the three commonly seen problems that when we try to express the eukaryotic gene in the bacteria cell, the intron, early termination signal, and the codon bias. And also this problem caused by the DNA sequence, all right? The DNA sequence that you try to clone. And this problem caused by the bacteria cells, all right? And number one is the bacteria cell might not process the recombinant protein correctly because some animal protein, okay, they are glycosylated. So that means they have this sugar group attached to the protein after the translation, and the glycosylation does not happen in the bacteria cell. So that could be a problem because, right? So the glycosylation. Secondly, the protein might not be folded into the correct configuration by the bacteria cell. Okay? Because, for example, the bacteria cell, they cannot form this disulfide, right? Disulfide. So disulfide basically is very commonly seen in the animal protein. Disulfide. So do you know what amino acid will give you the disulfide? Anyone know? So the audio keep cutting out. Let me see if this I can fix it. Okay, so basically the cysteine, all right, cysteine can form the disulfide bond and to stabilize the tertiary protein structure. Okay, so the bacteria cell they cannot do this disulfide bond between the cysteine, and also the bacteria might degrade the recombinant protein. All right, so these are the problems caused by the bacteria cell. Okay? And then via glycosylation. So basically, as I mentioned here, that because the protein might be glycosylated, the cell they cannot glycosylate the protein correctly. Okay? So this, and then you can use the eukaryotic cell to produce those proteins. So for example, cerevisia can be the host for a recombinant protein synthesis. And the yield relatively high, but also Saccharomyces cerevisiae cannot glycosylate animal protein, also cannot glycosylate the animal protein correctly as well. Okay. But another one here, these P. pastoris, all right. So basically, this one they can glycosylate the protein. Okay, so the P. pastoris, okay, which is the second species of the yeast, they can synthesize large amount of recombinant protein, and its glycosylation ability very similar to the animal cells. Okay, and then the expression vector for the P. pastoris use the alcohol oxidase promoter, which can be induced by the methanol. And the only significant problem with the pastoris is that sometimes degrade the recombinant protein before they can be purified. Okay, that's the pastoris. Okay, so the pastoris, they can glycosylate the protein similar to the animal cells, all right? And then for the Saccharomyces cerevisiae, several different promoters have been developed for the expression vector. So for example, the GAL promoter that can be induced by the galactose, and the 4,5 promoter regulated by the phosphate level, and CAV1 induced by the copper. All right, so these are some commonly seen promoter used by the Saccharomyces cerevisiae. And the Saccharomyces cerevisiae like an efficient system for secreting protein into the growth media. And that means the cell, basically the protein remain inside the cell, and that will create more problem to purify the protein. Okay. And the advantage is a safe organism for production of protein for use in medicine or in food. Okay. So that's the production of recombinant protein in Saccharomyces cerevisiae, and in the Pastoris. And then the filamentous fungi, the two most popular one would be the Aspergillus nigulans and the Trichoderma resei, all right? So basically they have a good glycosylation property, and also they can secrete the protein into the growth media. And this secretion is very important because you can purify the protein easily, okay? And so this is the promoter for Saccharomyces cerevisiae, the GALE promoter. We use the galactose and induce the transcription, the AOX, that's Pastoris. We use the methanol to induce the transcription. And these two, the glucomylase promoter uses starch to induce the transcription, and use the xylose to repress the transcription. Okay. And for the animal cell to produce a recombinant protein, okay, that can give you the complex and essential correct glycosylation structure. All right, that's why you use the animal cell for the recombinant protein production. Okay, and so you can use the insect cell as well. That's an alternative to the mammalia cell for animal protein production. And the insect cell right now is very popular as well, okay? Because they have a high yield of the recombinant protein. And the expression system basically is based on this virus, okay? And the farming, that's the recombinant protein from the live animal and plants. So for the use of silkworm for recombinant protein production is an example of the process referred to as the farming. Because you have transgenic organism act as the host for protein synthesis. So that's the farming. So the farming animal, so basically you have the oocyte, you remove the nucleus, so you have this cytoplasm, and then in the somatic cell, you will take out the nucleus, all right, and then you inject the nucleus to the oocyte, the cytoplasm, all right, and then implant into the foster mother. So that's the generational transgenic animal for recombinant protein production. So this is the farming in animal. So for example, in here, the recombinant protein production in the milk of a transgenic sheep. So you can have this recombinant protein secreted in the milk. So that's the farming, okay? So that's a general introduction of the expression vector. Any question? Okay, good. So the protein production. Okay. The production of protein is a long and painstaking process, and then we can split this production process into two different phases. One is upstream processing, and two is the downstream processing. The upstream processing, basically the actual expression of the protein in the cell, okay? And the downstream processing, that's basically the purification of protein and the verification of the protein function. Okay, so that's the two different phases of the protein production. So for the upstream processing, and for the first step that you have to select the cell to be as the protein source, and this cell we call it the host cell that can host the cloning, the expression vector, and then to express the protein. For the bacteria, the fermentation process are well understood and cultured in large quantity in short period of time, relatively easy to alter genetically, and can increase the level of production of bacterial protein by introducing additional copy of relevant gene to the host through the recombinant DNA technology. Okay. And then, of course, the most important thing is how is the gene is under control of expression, which we just discussed. Those are expression vector for the bacteria cell, right? So the most important feature is the promoter. So that and that is a promoter that can control the expression of the protein. And then the most commonly used bacteria species, we use the E. coli, right? And then also, of course, we discussed the glycosylation. That's very important, the glycosylation. Okay, the bacteria don't have the ability to add the sugar group to the protein. So that's the one important problem for the bacteria. So the advantage, okay, compared to the disadvantage that we discussed already. And then this is a continuous growth that I just show you, which I will show you the video now, the continuous growth. That's what normally people use in the field. Okay, so let me just show you the video. Let me just the video about the processing.
Many of today's bio
Can you hear the video very well? I'm going to play now. Can you hear? No, I posted. Okay, the video is not posted. Okay, so let me see.
Syllabus to the video. You can read it.
Can you hear? Just a second. down. Just one more time. Can you hear? Okay, just a second. Yes, that's the point that we have to show the video and then I will explain. But of course, the caption is also important. Just a second.